I am Joann DiGennaro, President of the Center for Excellence in Education, which I helped to organize in 1983 with the late Admiral H.G. Rickover, father of the nuclear-powered submarine and civilian uses of nuclear power. Our mission is focused and has remained in place all of these years to nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and math, and to encourage international collaborations between technologists and scientific leaders in the global community. Our programs are internationally recognized for their rigor, their excellence, and for producing some of the tremendous leaders in science and technology. We have 3 programs. You can go on our website at www.cee.org to learn about the Research Science Institute, the USA Biology Olympiad, and the Teacher Enrichment Program. With that, it is an honor and a privilege to introduce Michelle McMurry-Heath, MD, PhD, President and CEO of the Biotechnology Innovation Organization. Michelle is a medical doctor and molecular immunologist by training. She received her MD, PhD from Duke's Medical Scientist Training Program, becoming the first African American to graduate from this prestigious program. She spent 12 years working at the research bench before taking policy and leadership roles in government and industry. She comes to bio from Johnson & Johnson, where she served as global head of evidence generation for medical device companies, and then as vice president of global external innovation and global leader for regulatory sciences. Prior to her time at J&J, Dr. McMurry-Heath was also a key science policy leader in government. President Obama named her Associate Science Director of FDA's Center for Device and Radiological Health. She also was the founding director of Aspen Institute's Health, Biomedical Science, and Society Policy Program. It indeed, Dr. Michelle has had an illustrious career, which continues. She received her BA in Biochemistry and Molecular Biology from Harvard University. Dr. Michelle, we are so gratified to have you with us. I turn it over to you now. Thank you.
Thank you so much for that warm introduction, Joann. And it is a pleasure to be with you. I just want to thank the entire CEE community, the proud alums, your supporters, and the leaders of tomorrow who have tuned in. I wish we could meet in person, and maybe next time, as soon as the biotech industry conquers COVID, and believe me, we're working on it, we'll have the opportunity to do so. But you've picked an exciting time to explore leadership in the STEM fields, particularly in the life sciences. I'm not sure most of the general public understands the scale, scope, or speed of our industry's progress in just the past few years, but we are gaining scientific knowledge at a breathtaking pace. The ability to leverage genetic sequence data information was a transformative step forward. We saw that with some of the earliest vaccines for COVID, which were made in weeks without ever having to put the virus under the microscope, using genetic sequencing and synthetic biology. Now, of course, after those weeks, we've been testing them in animals and now in humans, and we're getting so close. These kinds of new breakthrough platforms helped the biotech sector do seminal work on two earlier coronaviruses, SARS and MERS, and that helped position us to respond so quickly to this one. We're a competitive industry, but this is really a time of unprecedented cooperation. We've seen amazing data sharing between companies and universities. Small biotechs and big pharma companies quickly entered into handshake deals and signed one-page brief MOUs or memorandums of understanding to get started. In Washington, regulators at the US Food and Drug Administration have never worked so fast or been so flexible. And I know this because I used to be one, as you heard. So government and industry both know how much speed matters. And we don't have the luxury of our normal 10-year drug development timelines to beat this pandemic. FDA deserves a lot of credit for embracing remote clinical trial monitoring during this emergency with so much apprehension that we have around traveling and around going out of our individual houses. It's one of the several innovations that we hope is precedent setting, even after this crisis is over. Small innovators and big biopharma companies worked together on manufacturing scale-up regimens for COVID that puts patients before profits. And many of them are manufacturing vaccine and drugs at risk before they even know if they're going to be approved or accepted. As a result, we've got hundreds of millions of vaccine doses already at the ready. And as a society, we can speed vaccines to people who need them the minute we get the okay from FDA. Then, of course, there's the social distancing, the shift work, and the long hours by those masked heroes and sheroes working in our labs. They've spent a lifetime studying and preparing for a crisis like this, using all that they've learned to end the pandemic and save lives, especially for the sick and the elderly. And that's the industry right now. In ways large and small, the worst healthcare crisis in a century has brought out the very best in our industry. And you can see that in the over 800 drug, vaccine, and antiviral development programs that we have launched against COVID just since the beginning of this year. And frankly, we're looking for brilliant young people with the courage and compassion to lead us into a future in which we can hopefully cure a lot of diseases. The topic of today's panel, how does a drug actually get from the lab to the market, was actually the question that first brought me to Washington over 20 years ago now. I started growing up in Oakland, California, the daughter of two public health officials, and really saw that the impact that science could have on vulnerable communities. At Duke, I had the opportunity to really immerse myself in the lab and loved working with science and working at the research bench. But then really had the opportunity when I had a year where I was breeding transgenic mice and there was not a lot else to do, to take some science policy classes. And I really got a different sense of how those science funding decisions get made and who has influence on which scientific questions get answered. And that's really something that has motivated me since then. I had a close family member who participated in an early clinical trial that turned out to be very successful, and he was in the effector arm, so he got to benefit from that early treatment. But it really showed me the challenges that families face when they're thinking about interacting and participating with medical research, and how much is on the line, as so many Americans this year have seen, when you're waiting on a cure to come from the research. I got to work with Senator Joseph Lieberman and passed a bill that helped redirect research dollars and to really that translational research that is so critical and so important to turning a great scientific idea or breakthrough into something that actually works for patients. And now at the Biotechnology Innovation Organization, which is a membership organization of over 1,000 biotechnology companies, big and small, ranging from biopharma to food and agriculture to energy and the environment. We really have had the chance to focus, since I joined Bio in June, on how we expand access to all this great promise that science is delivering. So the COVID pandemic has laid bare the clear links between poverty, environmental injustice, and race in our healthcare system. And this is something that we've seen in many disaster situations before, hurricanes and tornadoes. But this pandemic, largely multiplies this impact. We've seen, for example, that Black Americans are dying from COVID at a rate 3 times the rate of whites, and low-income individuals with underlying conditions are much more likely to contract the virus and have severe outcomes when they do. COVID is just the latest in a long line of diseases that show stark racial and socioeconomic disparities in outcomes. And one of my industry champions is a leader that has attacked this head on, a leader named Tony Coles. He's an African-American CEO, and he helped give me my start in the industry through a United Negro College Fund scholarship program that he started while at Merck. And it was through that program that I got to go to visit the Merck campus as a graduate student and see how the industry really takes these great ideas that academic science and NIH-funded research develops and turn those into real solutions that are practically and pragmatically impactful for individuals. That program had a powerful, informative impact on me as a young researcher in molecular immunology. And on June 1st, when I was honored to take the leadership reins here at Bio, it was the week after the George Floyd protests. And at our digital convention, I got a chance to do a panel with Tony, and he said something worth repeating. He said, we have an obligation to step into the vacuum of moral leadership in this country, because it's our birthright as leaders to fill that vacuum and to fill it productively with things that we want to see happen in society, because we cannot divorce the twin engines of social progress and economic opportunity. And for me, that means empowering young leaders to step forward, achieve equity, and lead our industry forward. And the purpose of CEE is to give you just the tools you need to do that. As exceptional scientists, you'll have many opportunities before you as scientists, as teachers, as policy leaders, and these are all noble callings, but I want to challenge each and every one of you to really consider the opportunities on the commercial side of our business as well. I want you to encourage you to learn about and utilize the resources that are available to you: incubators, accelerators, venture capital networks, mentorship programs, federal funding, and more. Biotech entrepreneurship is one of the most complicated, demanding, and rewarding career paths that you could possibly choose. But choosing it puts you in the driver's seat to really make the kinds of change that you want to see in our industry and in the world. And we need leaders that look like you, think like you, and most importantly, care like you, driving the biotech industry into tomorrow. So before I take your questions, let me just close with a dose of inspiration this morning. It's a true story of taking an idea to market and creating a more compassionate and equitable world in the process. Ted Love was in semi-retirement after a long and successful career at Genentech. He had a great family and had achieved a great career, but he felt a calling to use his accumulated wisdom to do what had never been done before, and that was to develop a medicine that goes after the root cause of sickle cell anemia. As you may know, malaria is one of the human race's oldest and deadliest diseases, and each year it kills millions of kids around the globe. And the parasite that originally caused the disease has been around for more than 100,000 years, giving natural selection lots of time to fight back. And in regions like sub-Saharan Africa, where malaria is endemic, the evolutionary response to this disease was the sickling of red blood cells into a sword shape that confers some resistance to malaria. Now, most people who are born with this variant have a sickle cell trait and may not even know that they have it because they only carry one copy of the altered gene. And why would they? They're healthy, their hemoglobin is doing its job, carrying oxygen to different parts of the body. But those born with actual sickle cell disease and that have 2 copies of this sickled trait face a difficult and uncertain future. They tend to have a shortened life expectancy due to multi-organ failure. They have excruciating pain episodes and can get an acute chest syndrome, jaundice, or strokes. For decades, modern medicine produced very little in the way of new drugs to help those living with this terrible condition. And sadly, it was for lack of trying. There simply wasn't the leadership or the capital to tackle this disease that afflicts people who are brown and Black much more commonly than it does others. But a man named Ted Love, an African-American scientist, did incredible work at Genentech, made the decision to lead. He took a promising piece of intellectual property and he combined it with his scientific know-how and his business acumen. And he started a company called Global Blood Therapeutics, or GBT. And he lived into his values, finding the best person for each position in his company, and created a very diverse company. He built a company with more women than men, a company where what we think of as sometimes as minorities comprise the majority of the workforce. And together they set out to raise the capital to translate that promising IP into a real solution for this devastating disease. Remarkably, TEG took the company public on the first 6 patients alone. And the data was so striking that the company immediately became worth $1.6 billion in an IPO based upon these 6 patients. And if you looked at the patient's blood under the microscope, you could see the sickle cells just disappearing. Last November, the FDA approved the first drug ever that attacks the underlying cause of sickle cell. GBT earned regulatory approval for Oxbryta on its very first attempt, 3 months ahead of schedule. And now Ted and his team are out there educating doctors, educating the community about this breakthrough and its transformative potential. And people who spent their lives enduring horrible pain episodes and other serious symptoms are really now getting some relief and a new lease on life. They're succeeding because the GBT staff reflects the community they're seeking to serve. And I hope TED's story inspires you as much as it inspires me. Our industry is ready for your inspired leadership. We are ready for you. So thank you all for the passion and your ambitions to be forces of social progress and economic opportunity. And I hope you take a big step towards knowledge, equity, and empowerment here and have an incredible and enriching experience with CEE. Thank you.
Thank you very much, Dr. McMurry-Heath, for your inspirational words and personal story. We're very delighted to have your invaluable experience and wisdom. For the question and answers, I'd like to touch on several topics of potential interest to our audience. These will be questions first related to your career path, second, questions related to drug development and path to market, and third, questions related to current developments in the industry, for example, touching on changes due to the pandemic. So maybe as we get started, first for the students who may also be interested in pursuing a career in the health sciences, what do you feel has been the most rewarding and challenging aspects of your path? What made you decide to go into your field of study?
Well, the most rewarding is always having an interesting and intriguing question to try to answer, whether it's a scientific question at the bench or a policy question about how do you try to attack something that has plagued society for quite some time and you're trying to come up with a new and novel solution. That kind of problem solving, as I'm sure everyone involved in this program would agree, is just one of the funnest things about science. And it really is a wonderful process. Challenges, I think, are not dissimilar from others. You know, you're always trying to find a way to fit that curiosity into life, to have some certainty about your path when actually your path sometimes chooses you, and you can't always predict where you'll be 5 or 10 years out. So I would say it's a combination of getting able to tackle those scientific questions and balancing the flexibility needed to keep pace with a career in the world as it changes.
Next, can you tell us more about your current position as the president and CEO of Bio? You know, for example, what is the day-to-day like, and what do you hope to achieve?
Well, I started at Bio in the midst of the pandemic. And so my day-to-day involves sitting on a lot of Zooms and really trying to connect with telling the story of Bio and of our industry. We've been trying to form a lot of partnerships and enable the work that our companies are doing around COVID, while at the same time advocating for them and enabling them to continue the work they're doing in so many other disease areas. Now, interestingly, Bio does not just cover biopharma, it also covers food and agriculture and energy and environment. So we've spent a lot of the summer really kind of revisiting our strategy and figuring out how we weave the narrative around the power of biotechnology across all of those factors and sectors of our society and what difference they can make. But I spend a lot of my day talking to people, understanding what issues they may face in their companies, talking to others outside of our industry about what we do, and advocating on behalf of the scientists whenever we can.
Switching to drug development, thank you for sharing the story that you mentioned before. In general, what are the challenges that you feel are facing the industry now in finding new drugs? For example, where have we been successful, and where might have we not been so successful?
Well, it's interesting because our area of biggest success is the science, right? I mean, we're in a year where CRISPR just won the Nobel Prize, and we have such amazing scientific techniques at our disposal that are really allowing us to answer some questions that, 5 or 10 years ago seemed unanswerable. So the science is no longer the rate limiting step. But often the regulation of the science, the distribution of the science, the payment for the science, the ability to have ongoing investment in the science, those are the things that can trip up our companies and our industry, and sometimes stand between the great ideas and breakthroughs that we have and the patients and the consumers who need them. So that's really why it's an interesting sector to be involved in the policy envelope of a sector, because it's one where the policy envelope is so determinative of what happens with the end result of the work.
Here's a question from our audience member. How do you see the roles of academic or public and corporate work in drug development in relation to one another?
Like two peas in a pod, they're both very necessary. Academic, curiosity-driven science is incredibly important. It gives us the basic understanding we need of the pathophysiology of disease, of different scientific approaches and techniques that might work to answer a question. It lets us understand the roadmap for example, in the human genome of where we need to go. But having trained in academia, there's sometimes a tendency to think that that's the bulk of what needs to be done. That's the hardest part. That's really what it takes to deliver a new solution to a patient. And yet, having had the opportunity to work with companies big and small, I can tell you that the science of turning those ideas into a practical medicine or drug that can actually help and serve a patient in a predictable way with a predictable amount of efficacy and an assurance of safety is its own complete scientific field. And you can't have one without the other. And often we find that academic part of the research is maybe 5 or 10% of the total development cost of bringing a new solution to patients, for example. So while it's critically important, it's foundational, it's not the whole shebang. And we shouldn't sell short the amazing innovation and scientific know-how that goes into that latter part of the pipeline.
So here's another question from our audience. How challenging is the policy discussion right now on CRISPR? How do you counter fears with potential scientific progress?
It's so interesting. We had the head of the Food and Drug Administration Center for Biologics address one of our meetings recently, and I asked him this very question. I said, you're in a situation where CRISPR is winning the Nobel Prize. It's the holy grail for many intractable human health issues like cancer, and yet when we talk about agriculture, it's feared and loathed. And he said, yeah, it's never made any sense to me that people say, yes, change my genome, but don't change the genome of my corn. And I thought that was just a nice way to put it in perspective, that the technology is the same and the promise of the science is the same. And we have to have a rational approach across all of its applications, and not be afraid of the science, but be in awe of what the science can unlock for us. Because biotechnology, for example, in agriculture and the environment is not a nice to have. It's a way in food, for example, to deliver highly nutritious, affordable foods to low-income food deserts in a way that could change the way children grow up today in the inner cities of the US, for example. So we can't have blinders on and be such purists that we don't see all the full potential of the science that's at our disposal.
Um, Thank you very much. For those, where do you think perhaps extraordinary students like those involved with CEE might best be able to contribute to kind of the industry and drug development in this endeavor overall?
Oh, there's so many possibilities. If there's any scientific question or disease area that interests you, there is a place for you and there is a chance for you to participate. There's, of course, very traditional R&D where you're working on figuring out how CRISPR actually becomes a drug that can be delivered to a cancer patient. That is an incredible portion of what we do. But then there's also designing the clinical trial so that you can test it and see if it works. There is understanding the regulatory process and putting together the applications that argue for the science and make it clear where the safety limitations might be. There's the epidemiology of following up on the use of the science to make sure it continues to perform and behave as you expect and hope. So there, as well as the entrepreneurial side of what we do, for which a scientific background can be incredibly helpful. So there's so many different applications for scientific excellence and achievement throughout our industry. And I hope you just explore a little and try it on for size and see if there's something there that interests you as well.
All right. So I think we have time for one more question. And it relates to perhaps issues about drug prices. Why do you think pharmaceutical prices have been such a massive issue within the past 2 decades? Could this be related to the increased difficulty in taking a drug to market in recent years? And what is your overall opinion on the issue? I mean, do you think it have a negative impact on innovation?
Yeah, so bio has a long tradition, long before I joined in June, of really advocating on behalf of reducing the cost sharing that patients share in paying for a drug that they need to take, decreasing, for example, the out-of-pocket costs that patients have to pay. That is incredibly important. But a lot of the growth that we've seen in the cost of pharmaceuticals has actually come from the number of people involved in the process. You know, there's lots of different sectors involved in delivering drugs to patients, hospitals, pharmacy benefit managers, health insurers. And so we all have a part to play in the solution, and it's important we all play our part. But that being said, we also need a holistic conversation about what it is we're paying for, because it's not enough to just have access to the solutions we have available today without also being sure that we're not slowing down the development of new solutions for tomorrow. Because I think there's very few people who would look around our hospitals and our distribution of disease today and say, we have produced enough science, that we have all the scientific solutions we need for patients as we're sitting here today. I mean, COVID is a perfect example that we don't have all the scientific solutions we need. And that means it's critically important that we develop those new solutions as quickly as possible. So we do not want to shortchange our future, which is so critically important, and finding those cures for diabetes, heart disease, stroke, you know, all of those areas that sometimes disproportionately impact poor and vulnerable communities, but do not yet have a silver bullet just to get access to the science we have right now. There's a lot more to come, and we want to make sure that we don't turn off the spigot of innovation.
Thank you very much.
And thank you for the invitation.
Appreciate it very much. Thanks again.
Take care. Bye-bye.
So I think for now, what we're going to do is to transition a little bit to a more panelists discussion. Let me briefly introduce myself and kind of a little bit maybe about what I do. So I was originally affiliated with CEE by participating in USABO in 2003. I started overall as doing biological research, even from as a high school student, actually, and have sort of continued that to where I am today. Right now, I'm running a startup called Stemcellarant, where our overall mission is really to help to do 2 things. One is to help kind of accelerate the early stage of drug discovery process, primarily preclinical stage, by making novel human cellular models from stem cells that can be used to help identify new drugs and new types of therapeutics. And we also have a second goal, which is also help make the development of stem cell therapies safer and more effective. And so our company is really focused on developing new technology. We do this by supporting as well as collaborating with many different large pharma companies, doing internal research, as well as partnering with other companies to develop cell therapies which can eventually be made into treatments for patients. Recently, what we have been doing is also been responding to COVID by developing human lung models, which can be used, for example, to study COVID and to screen for drugs that can be potentially therapeutic in nature. So I'd like now to kind of switch over and to introduce the panelists who will share their own experiences and expertise in different aspects of drug development. Our first panelist will be Dr. Lisa Purcell, and I'll let her take it away.
Thanks, Victor. I joined Regeneron in 2008. And so right now I'm working in our immunology and inflammation group in the clinical scientist role. I actually received, so I'm Canadian, from a really small town in Canada. I received a PhD from McGill, completed some postdoc training at Columbia. But really my current work focuses on the clinical development of therapies within immunology and ID, including those for SARS-CoV-2 and COVID-19. I also, I've been at Regeneron for a while, but I also worked on therapies just in general for pathogen in our preclinical space using some of our novel technologies. And part of what I do at Regeneron is also, it has to do with some of our students. So I'm the scientific director of Regeneron's science education efforts, including things like Regeneron Science Talent Search, some of the mentorship that we do, and also now our ISEF, Regeneron, and Regeneron's now the title sponsor of ISEF. So what we wanted to do in this panel was maybe to sort of introduce the stages of clinical drug development, because this is an aspect of things that, when you first get in the lab that you might not be exposed to. So funnily enough, I'm actually gonna pull up a slide, and I'm gonna stop my video, because if there's anything that I've learned is that I don't want to use up my bandwidth by having my video on at the same time that I'm screen sharing. So I'm going to pop up my slides here and just do a little intro to our clinical development. So obviously, when you start out in drug development, you really have to discover what you're working on. So scientists like Victor actually brainstorm how to target a certain disease or a condition. And the drug or therapy has to be made and then tested in the lab using usually cells and petri dishes. And then that drug has to be tested in animals, because you obviously have to make sure that something is safe and efficacious before you put it into people. Now, this stage can take up to several years. It can also be shortened to weeks or months, and we've seen this in the context of SARS-CoV-2, and this is especially true if the scientists are using platforms and methods that were already developed to target other pathogens or antigens in a similar way. So once there is enough preclinical data on the drug, that drug then moves into phase 1. And here, the objective is to show that the drug is safe and is typically completed in tens of individuals. This phase 1 can last days to weeks. And once that drug is shown to be safe, it moves into what we call phase 2, where the objective is expanded safety. Here, clinicians can also incorporate measures of efficacy of the drug. And typically, this is done in hundreds of individuals and takes usually weeks to months. Sometimes the drugs can actually move from phase 1 to phase 3 directly, and Mopley, who will be speaking a little bit later, will give an example of this. And finally, the drug moves into the late stage or pivotal phase 3 trials. And these trials typically take several years, and typically there's more than one of these trials completed. And here we look at thousands of subjects and incorporate measures of both efficacy and safety. And also not captured on this slide is that trials can be completed with a drug after it's approved if there's evidence that the drug could work in another disease setting. And Laura will talk a little bit about this a little bit later. The other thing that we wanted to highlight is that clinical testing typically takes years, and most drugs that enter clinical trials fail. So here you can see the approximate length of each phase of the trials. And then in the pink bar, you see an estimate of the percent of drugs that move to the next phase. So for the drugs that enter phase 1, only about 70% or 70 of 100 of those drugs move to phase 2. And then of those 70 drugs that move to phase 2, only about 30% or say 25 or so make it to phase 3. And then finally, only about 5 or 6 complete phase 3. And those data are documented and submitted to the FDA. They're reviewed and may be approved if the drug's benefits actually outweigh the risks. And it's only at that point that a drug is made available to the patient population. So I wanted to point out there have been many questions surrounding the clinical trial process for vaccines and therapies, especially in the context of SARS-CoV-2 and COVID-19. And I think it's really important to emphasize that Regeneron, along with other companies developing therapies for this virus and vaccines, are not skipping the steps outlined in those previous slides. Rather, the companies are working hard to really shorten the typical timelines by working faster to get the trials up and running, and then shortening a lot of the operational considerations within those trials. And so this keeps the development on the lower side of the timelines that I outlined on those slides previously. And I also wanted to mention that another way we're moving therapies through clinical development is by using adaptive trial designs. And so here, what's neat is that the trial is designed to address safety and efficacy measures for the different phases, and you address that in the protocol. So for example, the phase 1 safety trial is completed knowing the safety endpoints that need to be measured. And then once the enrollment is completed, you do an interim analysis that could on that data before you move to phase 2 and so on and so forth. So what's really neat about this trial design is it's completed with a set of pre-specified rules that happen before the study actually starts. And that outlines when and how the drug can move to the next phase. And so this design has actually been used for some trials of COVID-19 therapies. And the goal is to really make sure that a drug or a vaccine is safe first, and then quickly allow efficacy to be assessed in that context. Thank you very much, Victor.
Thank you, Dr. Purcell. Next up is Dr. Mobley Daws.
Hello, everybody. First of all, I'd like to thank everybody, thank CEE for the opportunity to speak today and be included on the panel. So my name is Mobley Daws. I'm an ID physician with a background in epi and public health. And I've been really interested in HIV since I was in high school, as HIV back then was the epidemic of our time. I've been lucky that in every step of my career, I've been able to increase the impact that I'm trying to have on the HIV epidemic. In the late 90s, I worked with a small community organization using flip charts to educate sex workers about HIV prevention. And then I was finally able to treat people who had HIV with combination antiretroviral therapy during my training and practice. And then I've worked also in public health to help prevent HIV at the population level and improve rates of virologic suppression in New York and San Francisco. And now I've been at Gilead for over 7 years, and I'm working on developing new medicines for HIV treatment and really the development of transformational new medicines for HIV prevention. My colleagues and I are totally committed to making sure everyone living with HIV has their HIV undetectable for their own health, and to make their HIV untransmittable, a concept known as U=U, and to make sure that everyone who's at risk for HIV has access to safe and efficacious prevention medicines that work for them in their lives. And we're also working very hard to find a combination of medicines that will help us find an eventual cure for HIV. The other thing I worked on at Gilead, like many of my colleagues, is we pivoted to helping develop remdesivir for COVID-19 for the past several months. So that's my introduction. And I wanted to talk a little bit about a drug that I'm helping develop for HIV prevention. And so Lisa, Dr. Purcell talked about the standard pathway for drug development. And our newest drug is a long-acting drug that can, lenacapavir, which is a capsid inhibitor. It's a first-in-class medicine, and it is given as a subcutaneous injection, like an insulin shot in your abdomen. And, she talked about how the preclinical phase can take a long time. So we've been working on this drug for almost 15 years. our clinical scientists, our research scientists found the first generation lead in 2006. They worked on it for over 2 years and then decided that this lead wasn't working. And then they screened thousands of molecules to find the second generation lead. And it took over 6 years to develop an appropriate clinical candidate, another 2 years to have our first in human phase 1 study, and then go into pivotal trials for treatment. And now we're planning the trials for prevention. So that's taken over 15 years. And I just wanted to highlight a couple aspects of this to talk about unique aspects of development for long-acting and for PrEP medications in particular. So the scientists who developed this compound never gave up. They continued to advocate for it because it had such amazing viral suppression activity. It's very, very efficacious at a small concentration, but it was super insoluble. And at the time that we started working on lenacapavir, the goal of the HIV field was to find a combination pill, a single tablet regimen that combined the entire cocktail into one pill once a day. And the characteristics of this drug were the opposite of what you wanted if you wanted to put 3 pills together into one small, easily swallowed tablet that's taken once a day. But as we were developing this drug, the field and what HIV treatment and prevention was looking for changed. We had developed a series of new combination tablets that got better and better from a point of view of safety, reducing the adverse event profile, and getting more efficacious and working better. But the drugs that we've been helping have helped a very significant number of people. And if you look at it from a population or public health perspective, places where there's high uptake of our latest drugs for HIV treatment and prevention can really change the shape of the epidemic and bend the curve to reduce the number of cases in HIV. But there's still people who aren't benefiting from daily oral pills. And so the field has moved to developing medicines that don't require daily adherence. Sometimes people don't want to take daily pills because they have trouble remembering, it doesn't fit in with their lifestyle. And other times people don't want to take them because they don't want anyone to know that they're taking a medicine to prevent HIV or treat HIV. So we started looking in the field at different delivery mechanisms, and then the liability of capsid, that it was super insoluble, became a huge asset. Because when you develop a long-acting medicine, you want something that's very insoluble and takes a long time to eke out and then lasts 6 months, which is how long lenacapavir lasts. So I think the lesson that we learned from this is if that something has really amazing activity and works well, to kind of stick with an exceptional molecule and figure out the way to bring it to people, and to really have our scientists and clinical development people connected to the field and what the field really needs, because those needs change over time. So something that was a liability can become an advantage and hopefully make a difference in helping us tackle and reduce the HIV epidemic. So thank you. And I will at this point turn over to Dr. Laura Bloss to take over.
Hi, everybody. First, thanks to CE for the invitation to participate in the panel today. My name is Laura Bloss, and I'm in Global Regulatory Affairs at Amgen. And I'll start with a brief introduction of my background. And then, as Lisa said, I'll move into some remarks about how we continue to develop drugs in the post-market setting, with a focus on the additional clinical trials that we do in the post-market setting. So in terms of a brief background, I have an undergraduate degree in chemistry and a PhD in biochemistry. After I finished my education, I joined the clinical development organization at Amgen. And for the first 12 years that I was at Amgen, I managed clinical trials, I led clinical development programs, and I also led the clinical aspects of regulatory submissions for new drug approvals in the nephrology, bone, and oncology therapeutic areas. After being in the clinical development organization, I moved to the regulatory affairs organization, and I did that about 10 years ago. In regulatory affairs, my current role is to oversee our interactions with global health authorities, our regulatory submissions, and our regulatory strategies across a portfolio of products. And currently, my portfolio of products includes products in the inflammation, metabolic, and endocrine disease areas. I'm also a department head at Amgen of our global regulatory writing and clinical trial disclosure groups. And that team, they prepare regulatory documents for submission. And these include clinical trial protocols, clinical study reports, and informed consent documents. And in addition, that team makes sure that we're compliant with regulations for registering clinical trials and posting clinical trial results on publicly available websites like clinicaltrials.gov. Maybe I'll say just a word about what I like most about working in the pharmaceutical industry and my job in particular before I move on to my remarks about drug development in the post-market setting. So what I like most about working in the pharmaceutical industry is really the fact that it's an industry that feels we can overcome any challenge. That is, as Dr. McMurry-Heath mentioned, nowhere has that been more evident in how we've approached developing drugs for COVID-19 and SARS-CoV-2. So it's a pleasure to work with people that are solution-oriented and have such a can-do attitude. In terms of developing drugs in the post-market setting, well, obviously drug development doesn't stop just because the drug is approved. After initial approvals in the US and Europe, we then embark upon getting the drug approved in other countries. And in order to do that, we have to comply with local regulations in those countries, which sometimes require that we have local clinical trial data to support drug approval, or that we do an ethnic sensitivity analysis. And the purpose of that analysis is to make sure that the results we see in global clinical trials can be applicable to that country's population. So that's an important aspect of drug development after a drug is initially approved in the US and Europe. And sometimes that requires additional local clinical trials in countries like Japan or China and other countries around the world. Next, drugs are usually developed for more than one indication, and clinical trials for these subsequent indications do continue after the initial approval. And I'll just give a couple of high-profile examples. The first is Keytruda. So Keytruda is a programmed cell death or PD-1 inhibitor. It was initially approved in 2014. Since 2014, FDA has approved Keytruda for over 20 additional indications and expansions of indications across a variety of tumor types. Another high-profile example is Humira. Humira was initially approved in 2002, and it's currently approved for 11 different indications to treat various inflammatory diseases. And those additional indications are usually developed after the drug is initially approved. Furthermore, if we have a disease that occurs in both adults and children, a lot of those pediatric clinical trials are conducted after a drug is initially approved. So thinking back to the slide that Lisa presented, we don't often follow that traditional phase 1, 2, 3 approach when we're developing a drug for additional indications in the post-market setting. Sometimes we go straight to phase 3, and in particular, we do this when we're investigating a closely related indication to an indication that's already approved, or when we have strong preclinical evidence that the drug will work in a new setting. And an example of this is for instance, a closely approved use would be, for instance, a cancer therapy where you're looking at that drug in an earlier line of therapy, or perhaps in combination with a different chemotherapy backbone. I should also mention that some of the repurposing of approved drugs that we've done for COVID-19 have gone directly into phase 3 clinical trials. And for instance, at our company, that was based on some strong preclinical evidence that we had that the drug could perhaps have a benefit in patients with COVID-19. Further, the pediatric studies in children also don't always follow that traditional phase 1, 2, 3 approach. And in the example where a disease is similar in adults and children, and we have a well-characterized relationship between drug exposure and response, we can sometimes go directly into phase 3 clinical trials to look at safety and efficacy of a drug in children. And what we do in that case is we use modeling and simulation to determine the doses we want to evaluate in children. And this helps us expedite drug development for children. And it's particularly important because those pediatric patient populations can be very small, which makes clinical trials difficult to enroll. And so I just want to end by saying, it's a great opportunity to have a therapy that can help people with many different diseases and serve many different patient populations. But obviously that's challenging. You need to make sure that when you're doing something like this in the post-market setting, you need to make sure that you're taking advantage of innovative approaches to expedite drug development, that you have the knowledge and expertise to apply those innovative approaches, that you have an adequately resourced program so that you can meet timelines associated with those clinical trials. And obviously that you evaluate emerging data from those clinical trials very closely and communicate new information to patients and healthcare professionals. So I'll stop there and turn it back over to Victor, who I think is going to take us through the Q&A period.
Thank you very much, Dr. Bloss. We have a large amount of audience questions, very nice, and look forward to discussing them with you in the next section. So before I jump directly into them, maybe I'll take a second to ask, to be a little cheeky and ask a question of myself to you folks. Maybe start things off and ask, do any of you have positions at your companies for internships for our students, perhaps, who might be looking for one right now?
Absolutely.
Yes.
Yep. So look on our websites. Gilead has an internship program, and we take students from college through graduate and in all different fields, from business to science to, a variety of backgrounds.
I think that applies to Regeneron as well. Probably.
Same for Amgen, and
Yeah.
our internship postings are usually early first quarter so that folks have time to apply before the summer.
And ours are actually up there right now. So you could just go to regeneron.com and take a look.
Great, thank you. I'll start with a question about kind of the perhaps estimated cost for taking a drug from early stage to market. Would some of you give a sense of like how much the projected cost might be for such a candidate?
That's a big one. Laura, do you want to take that?
Yeah, I can take that. Yeah, so, there's a variety of studies that look at this. I think the most recent estimates are around $1 billion for the whole thing. But, there have been studies that have put that number closer to $2 billion. And obviously that's an average. Clinical development programs, research programs are quite variable. Obviously, you can have drugs that serve a small patient population in a rare disease. Those development programs might not, probably don't reach the $1 billion. But then, of course, if you're developing a therapy in a much larger patient population, maybe a primary care patient population, a cardiovascular drug, then you're probably closer to the $2 billion range. So it's variable, but I think the most recent studies have been around $1 billion.
You think there could be a way to reduce the cost during at any point in the process?
So I think, Lisa talked about adaptive clinical trials, and I think one of our goals with applying these innovative approaches to clinical trials, like using adaptive clinical trials, I mentioned modeling and simulation in my remarks. Some of the reasons we do that, obviously we want to make sure that we're getting drugs to patients more quickly, so that's the main reason we do that. But it also has the effect of bringing down cost too.
Yeah, and that's really relevant in HIV prevention. We're at a point in the field where the traditional randomized controlled trial way that we evaluate drugs actually is not feasible anymore, because the drugs that we have work really well if you take them, but we need to develop drugs for people who don't take daily oral pills. So we've actually pivoted to a completely new design that can cut our sample size from 30,000 or 60,000 people that we would be required in the current environment down to like 3,000 or 5,000, which is much more manageable and helps reduce costs. But ultimately, our goal is to get it to people who need it as quickly as possible, but it has a side effect of reducing costs.
When a drug candidate, for example, is determined and they want to take it through a developmental pipeline, what are the considerations for determining, whether a drug will be selected for that subsequent treatment? And what are the parameters regarding the economics and feasibility outcome for determining whether to stop or continue development on a particular candidate?
Yeah, that's a big question. Maybe I'll take that, because it really depends on the indication. So I think there's a few common threads when you're in the preclinical space and you're trying to move that into human trials. And so there, what you're looking for is a drug that works. So first of all, you need to make the drugs, right? But you need to make a drug that's available, so bioavailable, but also, doesn't have any of the downsides in terms of, distribution within whatever tissue you're looking at, or you're able to take it orally if that's what you're looking for with the drugs. There's a lot of considerations. And then it actually has to work. So you have to show that it works in vitro, so in a petri dish in the lab, and then you'll have to show that it works in animals and that it's safe. And then you have to actually show that, it's distributed properly and it works like it should, and maybe some higher order animals, not always, but sometimes. And so there's a lot of stop gaps there. And, as a company or as a team, you need to weigh the risk benefit of not having one of those things and having the others, or going back to the drawing board and trying to make that drug better in some way to address something that maybe it's not doing well. And then you have to figure out, okay, what patient population that you're going to go into and what trial makes sense. Is it a healthy population in phase 1? Is it a disease population, depending on what that indication might be? And then once you're in the clinical development, you really have to look at safety versus efficacy, depending on which stage you're at, to figure out if you're going to continue with that. Is it working, and is it working well enough? Can you make enough of the drug? There are lots of other questions that come into play there. And they're all different depending on what that drug is.
Thank you very much. I want to thank everybody for participating. And I appreciate very much your coming in and introducing yourselves and talking so much about your work as well. I think I will hand it off to Misty.
Well, my goodness, it was so great to hear from each of you with your expertise. Our thanks again to Dr. Michelle McMurry-Heath, our panelists, Dr. Bloss from Amgen, Dr. Purcell from Regeneron, Dr. Daws from Gilead. I would be remiss if I didn't say a little bit about the moderator, who was chosen very carefully to help us out. Victor is a PhD, co-founder and CEO at STEM Celerant. He was involved with us with the USA Biology Olympiad, of which 10,000 students participate annually. He is an international medalist in the competition, so we are very proud of him. He is a developmental and cell biologist. He holds a PhD in biological and biomedical sciences from Harvard and his BS with honors in molecular and cellular biology from Stanford. Dr. Michelle King has helped out and prepared this excellent panel. And she and Dr. Lee will assist with getting answered questions from our participants because of the lack of time. So do send your questions forward to Michelle or Victor, and you'll get an answer. Now, I do want you, hopefully, to review the CEE website, and I encourage you to become involved with us should you have an interest in STEM education, our educational programming. This session will be available to review on our website. Of course, all our programs are free of cost to all teachers and students who participate. So I would be remiss as president if I didn't say, consider us this year for your donation to support STEM education. With that, be safe, be well, and thank you for your participation in this panel. Panelists, you were terrific, and we deeply appreciate it. Thank you.